Hi, everybody, and welcome back to Biology Bios. My name is Jim Edelman. I'm an associate professor in the Department of Biological Sciences at the University of Memphis. And my purpose with this podcast is really twofold. First, I want to let everybody know that the Department of Biological Sciences has really great research talks most Thursdays during the semesters, 4 p.m., Ellington Hall. People from all over the country and the world talking about the really cool research that they do. Also, I'd like to have those folks here on this podcast so we can learn a little bit more about them, how they got where they are, and what really excites them about what they do. And I'll say also that I did steal these questions from Rhett Miller's excellent podcast called Wheels Off, so please check that out as well. But without further ado, let's get to our guest this week. Thanks so much for being here. Well, Clay Cressler, welcome to Biology Bios.
All right. Thanks for having me.
So I generally start off with just a quick introduction, kind of asking you to say who you are, where you are, and maybe like the elevator pitch, like the short version of like what your interests are.
Okay. Yeah, so I'm Clay Cressler. I'm an associate professor at the University of Nebraska in Lincoln, Nebraska. I've been here for 10 years now, so I should probably stop telling people that I just got here. My work is really interested in understanding how processes that happen at one scale influence dynamics that happen at either a higher or sometimes at a lower scale. And I do that work a lot of times in the context of infectious disease. And so in that context, you might think about, how does what's happening within an individual, so the interaction between an individual's immune system and a pathogen, how does that scale up to influence things like transmission dynamics, and even ultimately the evolution of organisms. And so broadly, I'm kind of a host microbial ecologist, because my lab also does some work. We've started thinking about work about commensal organisms as well, and how those, commensal microbes might also influence processes like population dynamics. So broadly speaking, that's kind of where my interests are.
And correct me if I'm wrong, but when your lab's working on empirical systems, you guys are mostly doing Daphnia stuff, right?
Yeah, empirically, we do a lot of Daphnia work because it's a great system for that kind of cross-scale integration, because it's pretty easy to do individual scale experiments. We can measure immunity, we can measure different aspects of individual physiology, but we can also do really good population scale experiments and really see how things that happen at one scale up, and vice versa, see how the influence of things that happen at a population scale feed back to influence individual physiology. And that ability to work at both scales is actually not super common. A lot of the systems that are the best studied for individual scale things, like mice, for example, are actually pretty hard to do population level experiments. And things where you can do sort of population scale things like bacteria, it's a lot harder to do really good individual level things. And so in terms of that integration, I think Daphnia, like others like C. elegans or fruit flies, they sit in this really nice like sweet spot for thinking about cross-scale processes.
Yeah. I feel like the one example there that I think is really cool is the Drosophila one, the fruit fly example. Like, we think of it as like a workhorse in the lab, right? But those old Dobzhansky papers, they're like fundamental evolution things in the wild.
Yeah.
With this, I mean, there's so much potential there. It's really cool.
Yeah, and that's the other beauty with the Daphnia, is that they really do sit in this spot too, where they're C. elegans and fruit flies. They, like you said, they really have become lab animals. And while there are people who work on the natural ecology of both of those, it's not super common. And so sometimes the relevance of what's happening in the lab to what's happening in nature is a little less clear. But Daphnia have always kind of stayed pretty close to the wild. So, for example, like one of the projects that's happening in my lab right now is trying to understand how host microbe interactions influence individual tolerance to ingestion of harmful cyanobacteria, which are like the bacteria that cause harmful algal blooms, with the goal of kind of scaling up to saying, okay, how do those microbes influence the ability of Daphnia to like help to control a harmful algal bloom? And we can do work in the lab and at this kind of mesocosm scale and even at the scale of like ponds and lakes. And people do that pretty often with Daphnia. So it really has retained that, both a powerhouse in the lab and also still really ecologically relevant.
Yeah, absolutely. Well, all right. So that kind of like gets me to my next question. So you mentioned this one project, but you might not. I'm going to ask you to tell me about like one, maybe two projects that you're super excited about right now that you're working on, and without giving too much of your talk away, like why you're excited about those, and that could be the algal bloom one, or it could be something else.
Well, I could do ones that I'll just save the talk one for the talk, I guess, and tell you about two other projects. So the algal bloom one is a big one because we've been working towards that for a long time. When I first started my lab, we got interest. I had a graduate student who was interested in microbiomes, and so he did a lot of work just kind of figuring out who's there and trying to get some sense of how they interact with each other and how they interact with the host. And so it was really very basic, kind of like descriptive work early on. And then we started, as a kind of coming out of that work, one of the things that he did was this really cool experiment where he was looking at cross-generational effects of application of antibiotics, right? So if you take a mom, and some group of moms will never get exposed to antibiotics, and kind of look at their microbiome over time, and then you take another group of moms and you expose them to antibiotics, and then you take their babies, and some of their babies get to live not with antibiotics,
Right.
and some of them will stay in antibiotics, and then you just continue that for a while. One of the cool things we found with that was that we were not expecting at all was that over time, even in the control treatment, the microbiome got simpler and simpler and simpler, which is basically just like an ecological bottleneck is what was happening, right? Because there's like a narrow, they acquire their microbes horizontally. So basically, mom is like seeding the water with her gut microbes, and then when her babies are born, they pick it up in the water, and then that's how they assemble their microbiome. This is the thing that we worked out early on. And so when we were doing these experiments, where as soon as the babies were born, we were very quickly like taking them out of the water with their mom and putting them into a vial by themselves. So that we only gave them this really short window in which to acquire microbes. And over time, they basically just acquired the most abundant things.
Right.
And so the microbiome got simpler and simpler and simpler. And, but what we found that was weird about that's sort of, okay, that makes sense when you think about it. But what we found that we weren't expecting was that as the microbiome got simpler, the individual fitness went up. So they did better and better and better over time in terms of longevity, growth rate, reproduction, which we weren't expecting, because the paradigm in microbiome work is that diversity is good.
Yeah.
Having a diverse microbiome is better than having a less diverse microbiome. So we started on a series of experiments to try to figure out why. Why is a simpler microbiome better? Is it just that the most common things are also the most beneficial, in which case, why hold on to anything else? Because the cool thing, one of the other benefits of Daphnia compared to even like fruit flies for microbiome work, is that even in the lab, Daphnia retain a very natural looking microbiome.
Oh, I was going to kind of follow up with that. Yeah.
Yeah. It's crazy because fruit flies don't, right? So fruit flies end up with this super weird microbiome. It's very low abundance. So if you're trying to do DNA sequencing of fruit fly microbiomes, it's often a big pain because they just don't have much. Whereas a wild caught fruit fly will have tons of microbes. But Daphnia are not like that. Their microbiome looks very similar to a wild caught Daphnia. All the same groups are there. So what they're holding on to in the lab, for the most part, unless you do this weird bottlenecking, looks a lot like what they have in nature, which suggests it's valuable. It should be like have some function. So we embarked on this like series of experiments to try to figure out, well, what are these rarer members doing? Why do they maintain them? if they don't do anything. So we thought, okay, well, maybe it's something that you only need it in a bad environment, because we were raising these Daphne in an awesome environment. Like maybe they just need, the most abundant thing is awesome for them because they're not being challenged by anything. So we tried a whole bunch of different things. Exposure to a bunch of different kinds of pathogens, starvation, heat stress, cold stress. No, we never saw any sign that any of these, it was always simpler was better for everything. And then we tried an experiment with microcystis. So we exposed them to this toxic algae. Microcystis is the most common cyanobacteria that forms blooms. It produces a toxin called microcystin that's really harmful to most eukaryotes. So we did this experiment with microcystis, and all of a sudden we saw something. Now all of a sudden those certain groups, rare groups, seemed like they made a difference to the individual's ability to tolerate ingestion of cyanobacteria. So basically to maintain fitness, even when they're being fed a diet that was like 50-50 by sort of weight of microcystis versus like healthy green algae.
So like functionally in the wild, to be able to survive one of those blooms, still have offspring.
That's the hypothesis.
Yeah.
So this has been a long gestating project that I have a grad student who just ran a big mesocosm experiment this summer looking at the role of microbes and sort of microbial dynamics in the face of harmful algal blooms, but also eutrophication, and trying to tease apart what's happening in terms of the water microbiome and the Daphnia microbiome and the population level consequences. So that one has been kind of that's a project that really has been in the works for almost a decade.
Wow.
Just sort of slowly chipping away at various parts of it. So that one's one I'm really excited about.
So could you also talk briefly? Because all right, full disclosure, I know Clay fairly well. We've been working on some projects together, and that's the reason that I invited him out to give a talk. Well, that, and he does very cool work. But that's how I know him. But what you haven't mentioned much is that you sit at this interface between theory and empiricism. Like you were just, if I just heard that right now, I'd be like, oh, well, this guy, he gets his boots money. He gets critters in ponds and he puts them in cattle tanks and he looks at what they do. Maybe not cattle tanks. You probably don't need something that big for Daphnia, or do you?
No, we do. Yeah, no, we
okay, you do.
Tank.
Okay.
Yeah. These like the 2.5 meter tall cattle tanks, basically.
Nice. All right. So with that, though, you've also got all this background in mathematics, and a lot of your work sits squarely in theory. So can you kind of talk about straddling that divide?
Yeah. So the empirical stuff was things that developed really as a postdoc, actually. And then, as I've had a lab, it's often a lot easier to get students who are interested in doing empirical work in a biology department than theory work. But my background is entirely mathematical, actually. So my undergraduate degree is in math. My PhD work was entirely theory-based, doing like life history, evolution things. And so that's kind of home for me, is to kind of think about projects from a kind of mathematical perspective. And so really like the empirical work is all stuff that I've developed much more recently. And it's because I think the best kinds of theory are the theory that can tell you something about what's happening empirically. And that's kind of why I like the cross-scale stuff, right? Because mathematics provides this way of making hypotheses about how something that happens at, say, the individual scale will scale up to influence the population scale. This is a thing that's kind of hard to do just purely empirically, because you can often talk yourself into kind of anything you want. And it's reasonable, totally plausible kind of things. And so math in that context can help to constrain the universe of the possible, and define the contours of the plausible. And I really like that as a tool. So a lot of my work, what we've been talking about, a lot of the work that I do, what I'm going to talk about in my talk, is work where I'm sitting kind of more in that that collaborative space where what I'm providing is kind of theoretical background to help make predictions, to help frame how we understand the world. Because I think, you know, math is just a very precise way of making a hypothesis.
Yeah, yeah, that's super appealing. I mean, one of the reasons I like collaborating with folks like you is exactly that. And you have the chops to do it, whereas I actually don't have the chops to do it. I have the chops to sit in a room with you and be like, that is very cool. Like,
I think that's actually an underrated skill. I think not everybody can do that though. To understand the way that that theory. I mean, lots of people work in a really like theory-free way
sure,
and can make
sure.
tons of really important progress on problems even without kind of thinking about it from with any kind of theoretical framework in mind. But I think I love working with empiricists who couldn't do the theory themselves, but they totally get why that theory could help them. For me, that's awesome because you guys are always way better empiricists than I could ever be. And so it makes me feel like I have something valuable to contribute.
I mean, I feel like there's like a lesson for folks that are like just getting into science or whatnot, that like so much of it's collaborative, right? Like
Yeah.
you don't necessarily have to be the best at everything. Like you have to find other people that are really good at the things that you're not as good at, and then you can do these really awesome projects. Like,
Yeah. And that's the other beauty of theory from my perspective is theory is kind of simultaneously about everything and nothing. So it creates a really awesome opportunities to collaborate. Like I love
mhm.
the fact that, you know, one day I can be talking with you about birds and conjunctivitis in birds, and then the next day I'm talking with a collaborator about mouse immune systems, and then another day I'm talking about, you know, guppies and their ectoparasites, and all of that is, you know, it's all science, right? It's all work that gets me excited and happy that I have this job.
Nice. That's a very great segue to my next question was, speaking about being a scientist, um, was there like a moment that you knew I want to do science? Or was it a slow burn? Like how did you arrive at that?
Yeah, I think I'll give a two-part answer to that. There's the sort of like, how did I know I wanted to do science? And then there was the, how did I know I wanted to be a professor? Maybe what might be two
Cool.
different
Yeah.
things. So the, how did I know I wanted to do science was when I was, as I said, I was a math major. So one summer at my undergrad, I did a dynamical systems theory class over the summer. And the final part of that class was you had to pick a project and it didn't have to be biology, it just any kind of dynamical system. So, you know, some people in the class did like traffic flow and modeling traffic flow and the way traffic jams develop. But I had just gone to Yellowstone National Park the previous summer, and it was right after they had reintroduced gray wolves.
Yeah.
And so we had I was there with a group of kids. Basically, we were all like high schoolers. And we'd talked to the park rangers, and they were saying, like, hey, let us know if you see a wolf. You probably won't, but if you do, let us know where you found it because we're curious how they're going to use the landscape. We're not sure what they'll do because they haven't been here forever. And now, of
Right.
course, you know, everybody knows about wolves in Yellowstone. It's this great ecological restoration story. But in 1999, it was still totally unclear what this was going to do.
Yeah.
Um, so so I remember that thinking they didn't know, right, that the people that we were talking to didn't know what was going to go on. And so we had done a little bit of predator prey theory in the class as an example of a dynamical system. So I built a model for the interactions between wolves and elk, but also coyotes, which had become the dominant predator
hmm
and would interact like not in a competitive way with wolves, right? They don't eat each other, but they do compete for the same food. So it was a very simple kind of, now I know, didn't know at the time, you know, kind of Bob Holt kind of theory project. And it was so much fun
Nice.
because I got to spend all this time reading papers and things about wolf behavior and coyote behavior. and trying to figure out like how will these things interact with each other. So I did this project. It was lame. Honestly, it wasn't like the coolest project. The results were, whatever they were. I can't even totally remember what they were, but I gave them a poster presentation at an undergrad research symposium, and a biology professor came up to me and said, and talked to me about my poster. And then he said, you can do this in graduate school. And I remember my follow-up because that was literally I don't know why this is stuck in my head, but he said, you can do this in graduate school. And my follow-up was, what do you mean by this? And what's graduate school? Because I didn't know. I'm a first gen college student.
Yeah.
So,
no, I think that's not an uncommon experience, actually.
Yeah. So I had no idea that, I could do. And so he was like, well, you should come and talk to me. And so I went and chatted, and it turned out there was a professor in the math department that he was collaborating with, trying to get this math biology program kind of started up. So totally serendipitous that somebody informed me that math biology was a thing that existed. And I was so naive. I remember interviewing for graduate schools thinking that I was going to thinking specifically about like modeling evolution and thinking nobody's done that. I'm going to be the first. And then I realized like, oh, right. Yeah. Except for like Fisher in 1920. Right.
I feel like that was a lot of grad school for me. It was like, I've got this fantastic idea. Then I'd go to the literature and be like, okay, that was answered in 1930.
Yeah.
And then like slowly you're like, oh, well, that was only answered in 1982. Like I'm getting better. And then like after a while, you're like, oh, wait, this one actually hasn't been answered, but it takes all of that. Like, you have to go back through that.
Yeah. And I think that's the arc of graduate school is you come in thinking you have all the problems, exactly what you're going to do because nothing's been done. And then you go into the low ebb where you're like, everything's been done.
Mm-hmm.
And then you learn enough that you realize, oh, wait, no, actually nothing's been done. Like we've just scratched the surface of everything. And you go back, it's like, now I'm like, I have too many problems where like third year of graduate school, I was just like, I have nothing to contribute to science.
That low point is like super common. I don't know. I feel like people do talk about it. But like it's so ubiquitous.
Yeah.
Actually, you kind of got me to my next question, right? Oh, but you wanted to say about the professor. Yeah. Let's do that first.
Okay. I'll do that really quick. Cause I think there's a good sort of lesson in this. So when I was in graduate school, for several years, mostly because nobody else wanted the job, I coordinated the seminar, which I didn't coordinate who was coming, but I coordinated the graduate student lunches with seminar speakers. So for years, I was like just having to like wrangle, getting people to come to the lunches and talk to the speakers. And sometimes it was super easy. We'd have somebody that everybody knew and everybody would show up and that was easy. But sometimes there were definitely weeks where it was just me. And I was having lunches with these scientists who were doing really different things because, it was a big department. We had people all across the, speakers from just kind of every possible part of kind of biology. Really, it was kind of ecology and evolutionary biology, but still, that's a big tent of thing. So there were definitely weeks where I really had to sit for a minute with somebody's like CV and try to figure out what am I going to talk to these people about. And by the end of the first year doing it, I realized I love this. It's so much fun to talk to people who do stuff that's totally different, to try to figure out like, what can I say? Like, how can I relate what I'm doing to what you think about? Like, how are we going to have a conversation? And realize it's not that hard, actually. Yeah. And it's often really fun and interesting. And you'll learn something new, but also like think about your own work in a different way.
Very cool.
And so I think, once I sort of realized that, I was like, this is definitely what I want to do, right? I want to like keep having the opportunity to have these conversations, to meet people who do different things and just think about science. It's just so much fun. I couldn't imagine doing anything else.
Yeah, that's one of my favorite parts. I mean, one of the reasons that I think that this podcast is worth doing. I mean, I hope that we're going to have more and more undergrads that will listen to this and become engaged and hopefully come to your talk. But like, at this more sort of personal scale, like I just get to talk to people that do cool stuff that I don't think about all the time. And
Yeah.
and then it frames your ideas differently. And even if it doesn't like affect your work directly, it just kind of it affects your worldview.
Yeah. Yeah, absolutely. I think and it's just good practice too. I mean, for anybody who is already confident that they want to go and be a professor, forcing yourself to sit in meetings and have conversation with people who do really different things is such good practice for job interviewing, right? Like
Mm,
when I got to job interviews, like much later, I wasn't as nervous about talking to somebody who was a virologist or a computational phylogeneticist or whatever, because I was like, I'm sure we can find something.
yeah.
There will be some bit of common ground where we will both go, oh, we're both interested in that.
There's kind of a nice thing with biologists too that like most of us like yeah we're question driven about the things that interest us, but like we also either really like critters or really like plants or really like fungi or something, like there's just this like natural history thing that we're like, oh, isn't that cool?
Yeah, right. Yeah, I think everybody has a like, ain't it cool thing about their
Yeah,
system that you can always connect with.
yeah, absolutely. um okay, so here's where I want to go next, because you talked about that low point in grad school, and I feel like that relates to this next question, which is so much of what we do gets evaluated from outside. We submit papers, we submit grants. People say this is good about it, this is not so good about this. Sometimes they say it nicely, sometimes they don't. Um, but we also have those voices in our own heads that are like, oh man, I came up with this idea. No, that idea is not good enough. That's not going to fly. And we can all like shoot our own ideas down.
Yeah.
Um, how do you like deal with that voice when that voice pops up in your head?
Yeah, that's a good question. I don't know if this answers your question totally, but I'll go with it because it is kind of how I've dealt. I think this is sort of how I deal with it, which is in some ways ignorance is bliss. So oftentimes I start projects without doing a super deep dive into the literature, because I think a lot of times that's where that voice comes from. Like we were talking about in graduate school, right? Is you start
Fisher did
reading
it in
papers because you're taking
1927. Yeah.
classes, and
Yeah.
your advisor's telling you read, 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 which is it's great, especially early on. But it does create this intimidating feeling that everything's done, everybody's had all the ideas, you feel like you don't have anything to offer. And so one of the things that I have kind of done now, and maybe it's good, maybe it's not, is I don't read a lot until I'm done, or until I reach a point where I'm stuck, or and that way I kind of, because a lot of times what you find is even if somebody's had kind of the same idea as you, it's not exactly the same, and there's still something that will be learned from if you just push through with whatever it is that you're working on. And this is a little easier for me to think about in the context of like theory, right? Where I
Mm.
have a theoretical model and I want to analyze it. It's a little harder to think about that. I appreciate that it's not the same for thinking about like an experiment, but I think the same
I mean, yes and no. Like,
applies.
there's different flavors to it, but I do. I agree.
Yeah, because oftentimes if you get all the way to if you think about what other people have done right at the get-go, you can talk yourself out of an idea. Whereas if you look at it after you've finished it, you'll realize you didn't exactly duplicate what somebody else had already found. What you found is complementary to what they found, but adds some new dimension of understanding to it, or you have a different way of thinking about what it means, what the results mean, than what somebody else found. And so, like, keeping yourself a little bit free of the preconceptions that you can get when you just try to read everything that everybody's ever done. For me, I find to be valuable. It probably doesn't work for everybody. And I totally can appreciate why that wouldn't work for everybody. And then if I ever do find something where I do a thing and I realize I get it done, and I'm thinking, okay, now I need to go and find out what everybody knows about this. And I find that it's already been done to some extent. Like, yeah, that's a bummer, but it's also kind of like, oh, it was a good idea, actually. That was a good idea
Yeah. Somebody
because
else had it.
somebody
Somebody
else
else thought
had
it
it
was good.
too. I'm not an
Yeah.
idiot. Like, I came up with a thing that somebody else also thought was worth investigating. And I think that even that kind of reframing of saying like, oh, it's already been done, that's a bummer, is one way to look at it. But another way is to say that's a total validation that you had a good idea in the first place. And if you had one good idea, you could have another one.
I like it a lot. Yeah. That's a really nice way to frame it. And I think if folks haven't been there already, and you're in grad school or you're in an undergrad research situation, like you'll probably get to one of those points. Yeah, that's really so actually, all right. This has been actually flowing really well with my like list of questions here.
No,
Like I was
that's
like, okay, this is a good transition. Um, so thinking about from an advice perspective,
yeah.
what advice would you give like an undergraduate version of yourself? Like, so, a 20-ish something Clay Cressler, but working in today's world of biology, of science.
In terms of like just anything,
It
anything.
could be totally anything.
Okay. Yeah. I mean, my first advice is to everybody is developing some quantitative skills will never hurt you. Um, that can be bioinformatics. Obviously, I think that's a huge. It has become huge and will continue to be an enormous part of science going forward. So having at least familiarity with what the tools are and what they do, it will be valuable for anybody who's just trying to read the literature in now and especially in the future. I think.
Mhm.
Um, I would also say a class in like mathematical modeling is valuable, especially if you can find that where it's pitched at the right level, where it's really about. I think the value of math for me is more about the way I think about problems rather than it's a tool set for sure, but it's also a way of thinking, and I find it very valuable to me to be able to think like a mathematician sometimes and not always think like a biologist. Mathematicians, one of the power of mathematics and the way of mathematical thinking works is it's constantly in a simplification. Trying to boil a question down to its simplest possible form, right? But biology is complex. And I think the innate tendency so if I ask a class of undergraduates, okay, we're going to think about um, modeling the spread of an infectious disease. What are all the variables and the processes that you think we should keep track of? And they can go for 30 minutes just giving me
Yeah.
totally plausible variables, right? Things that absolutely could matter and totally biologically viable processes too. And then you say, okay, how would we put all of that into a model? And you go, oh, yeah, that's a lot. It's a lot. Like, yeah, but okay, now let's think about it like a mathematician. What are the key things? What's the minimal set of stuff that we definitely have to have? And I think that getting facility, not necessarily with modeling per se, but with modeling as a way of thinking can be really valuable.
Yeah.
I guess the other thing I would say, which is almost contradictory to the first thing about bioinformatics, is to say that there's still huge, huge value. I mean, one of the things I love about your work is that you're still out there beating the drum for experiments.
Hmm.
And I think, especially in certain areas of biology, there's this big move towards, well, we have all of this data. We need big data integration techniques and all of these, like, oh, let's throw, especially with AI becoming more powerful and machine learning techniques and things that allow us to process huge amounts of data into complex statistical models and things like that. That's all great. And as far as it gets us, but I think fundamentally, we answer questions in science with experiments, and always remembering that I think is really something I would emphasize to anybody, even the people who are like, I want to go get a degree in bioinformatics or something, is to remember that, applying those bioinformatics tools to well-designed experiments
Yeah.
is always going to be way more informative than mining a huge database. That's my opinion. Anyway.
I mean, I tend to agree that like, I mean, we get so much out of those sort of monster data sets. So
Yeah.
definitely don't want to dismiss that. But like, just in my experience, like when I've designed a really good experiment, my stats are a lot easier to run. When I
Yeah.
haven't, like it's a pain on the back end.
Right.
Um,
Yeah. Yeah. And your inferences are always so much stronger, when you know what you've controlled and what you haven't. And yeah, I love a good experiment.
nice. And thank you for those flowers there. I appreciate that. That was um so just really two questions left here, and I'm gonna just kind of combine them because
Okay.
I like to hear a little bit about like what people do as like work-life balance. Like what do you like to do when you're not sciencing?
Mhm.
Um, and then kind of within that area or anything else, like are there other things that you wish I had asked you that I didn't ask you?
Okay. Um, so when I am not in the lab, when I'm not doing science, I actually got involved a few years ago when we moved to Nebraska. We moved from Canada to the United States. My son was 13 when we moved, which is
Oh
a
wow.
tough age to
Yeah, it
move
is.
from to move to a new country. And he felt very dislocated by that. And so we were kind of desperate to find something for him to do, where he could meet people and make friends. And a friend here said, "Oh, there's a Boy Scout troop that meets around the corner, literally around the corner from the house we were living in. And I know the guy who runs it. He's really great." So we were like, okay, that sounds good. I was a scout when I was a kid. Um, and so we took my son to it. They were super welcoming. They open arms, and he immediately like made friends and had this community. And then slowly but surely, we've kind of the whole family is now roped in. So I'm assistant scoutmaster for a troop in Lincoln. It's boys and girls because it's not Boy Scouts of America anymore. It's Scouting America now, and
Cool.
we are combined unit, so co-ed unit of boys and girls. So my daughter was also involved. They both were Eagle Scouts, and
Nice.
yeah. So every week I am at scout meetings. I took 33 people backpacking to Colorado this summer. So that's kind of what I do within the troop. Is I run the high adventure program, so backpacking, whitewater rafting, rock climbing. When we want to do
Nice.
trips like that, I help organize them and lead them. So that's kind of my big thing outside of the office that I
Very
do
cool.
sort of stay balanced a little bit.
Yeah, absolutely. And yeah, like what else what haven't I asked you?
Well, so sometimes I get a lot of questions about how did you end up as a biology professor with a math back degree, which is a weird one, because I went into graduate school in a biology graduate program without having taken a single biology class.
Wow.
Yeah, which was weird. But what's one of the cool things about biology is that biology doesn't care. It's a big tent discipline. And so when I was applying to graduate schools, I applied at almost exclusively math departments, except University of Tennessee, where all the people doing cool math ecology were in the ecology and evolutionary biology department. And when I interviewed at all the places, I just loved Tennessee. I thought it was amazing. And I liked all the people that I was going to be part of my cohort, and all the other graduate students. And they were willing to take me. And so I went, even though I had not taken any biology. So I spent years, actually, the first several years of graduate school, just trying to figure out what was going on with biology, just basic biology stuff. Like to the point where when I was here, faculty, this is going to sound really bad, either bad or hilarious, I don't know, maybe both, I was teaching introductory biology, and we were doing the chapter on, we were doing the unit on endosymbiosis. And I remember reading, like looking through the book and developing the slides, and I went, what? Mitochondria were like a whole other organism? They have their own genome? Like, are you seriously? I just didn't know. I had no idea.
The way that you said that, though, like, even having known that for years, like, that's bloody cool, isn't it?
Yeah, it's so cool. That's what I love, right? Like, if I had gone into a math department, what would I teach? I'd teach calculus. There hasn't been much exciting developments in calculus in a while.
I think Newton got a lot of it right?
It's been pretty much taught the same way for the last several hundred years. But biology is awesome because we learn new things all the time. So every year I learn something new, and I want to bring it into the classroom and share it. And so I'm still, I think in some ways, not having much of a background in biology makes me constantly surprised by things that, first year undergrads know, and I'm still learning and am surprised and fascinated and captivated by all over again. So
that makes total sense. And it, that sort of excitement is infectious. So I feel like that's a good thing.
Yeah, I think so.
Awesome. Well, Clay Cressler, thank you so much for chatting with me today.
Thank you, Jim. That was awesome.
We really look forward to having you here in a couple weeks, and we'll talk then.
All right, looking forward to it.
A big thanks to Clay Cressler for a really fun interview there. You can hear more about Clay's work Thursday, November 12th, 2024 at 4 p.m. in Ellington Hall, and I hope to see you all there. Thanks for tuning in.